Okay, I think we're going to go ahead and get started. So I'm going to introduce Coen here, and then I know people are still filtering in. Um, but it's my pleasure to introduce Coen Elemans. I've known Coen for a while now, ever since he was sort of leading a big collaborative grant on the function of muscle in vocal communication, which is, I think, his specialty area, broadly speaking, studying the neuromechanics of sound production. And I think this is a really cool topic that is very related to IMSI, but is a very different from what we normally are talking about with locomotion studies. And Coen did his PhD in Wageningen in the Netherlands, and then did his postdoc in the US at the University of Utah. He's now a professor at Southern Denmark University and has won a number of awards, some of which I can't even pronounce, Coen. So thanks for that. And I've been visiting his lab a few times while I'm out here on sabbatical, and it's just been very interesting to sort of see him taking a very complementary approach to what I think many of us are trying to do in the MZ community, but in a very different system. And so we wanted him to highlight that and give a talk to us about the cool ways that he's thinking about vocal sound production. So Coen, oh, and in his spare time, he apparently goes out and dissects out the vocal folds of sperm whales that have washed up on the beach here. That's what he was doing last weekend. So, all right, take it away, Coen.
Great. Well, thanks so much for inviting me. It's really a really great pleasure to talk to this community and a lot of people I know from papers and from a long time ago at different conferences. So it's really, really cool. Good. So before I start, I'm going to take a whole step back and talk a little bit about sound, just to get everybody on board. And basically, sound is a disturbance of pressure that travels through a medium, right? So if you see the red dot here, it sort of travels around in an equilibrium state. But sound is made by movement, right? So as soon as animals started to move about 500 million years ago, there was information in sound about where animals were, like for predators, but also for prey, of course. And so sound is basically everywhere. It's this constant information stream that you can tap into. And very quickly it became crucial to be able to detect that sound for survival. And so we've seen that like evolutionary trajectories over lots of different animals have been shaped to detect sound. And even single cell organisms are able to detect sound. Good. And then something really special happened about 200 million years ago. This is a reconstructed sound of a Jurassic bush cricket. And basically, this is the first evidence we have of an animal deliberately making sound for communicative purposes. And so sound became crucial to communication. We think around 200 million years ago in bush crickets, but also in the vertebrates. And since then, it's and basically we see. I never know how long this clip goes. I'm sorry, but basically we've seen that like sound has been evolved in all these different vertebrates, and has become crucial for their survival and in lots of different ways. Good. And then something other really special happened. About 40,000 years ago, humans evolved language, which became absolutely crucial to our cultural evolution, but also to our ability to completely dominate the planet, right? Without vocal communication and language, we would have never been able to become the dominating species. And so sound production there has been super important. Now, and since then, humans have done something else. We've been taking care of our planet in a rather poor way, and we've been heavily polluting our planet in many ways, but also with noise. And so, so I'm trying to say stuff now. I'll go back to that slide. I tried to say stuff, but you can notice that the noise is just masking my voice. And depending on where you are, so if you would be rather close to me physically, you could still understand me fine. But if you're further away, it would be more and more difficult. And so one thing everybody agrees on is that the effect of noise pollution is that it really decreases the communicative ranges over which animals can communicate. But it also has massive disease effects. So we know at least, like in Europe, for example, 60 million people on an annually live in an environment where they basically live in a noise environment above thresholds for so they could get sick. There's at least 12,000 yearly deaths on heart diseases in newborns that are related to noise. So noise is really a problem in our daily environment. And that's where we know about it. And look, if you look underwater, it's even worse. We're just not so, we don't experience this on a daily basis, but the amount of noise we produce on the water is absolutely insane. And it's really affecting the communicative ranges of basically all the marine mammals and fishes and so that live there. So this is a noise map just of shipping noise that of currently on our planet. And the red lines are basically the major shipping routes. So you can see there's lots of noise in a large chunk of the planet. That's shipping noise. Then there is seismic air guns that we use. There's basically big, huge detonators that hang in large arrays behind boats, and we use those to send sound into the sediment and then look into the reflections of and try to analyze those for different sediment layers and look for oil and gas, for example. Good. And then there's also pile driving. So lots of activity in these marine environments causes noise. Now, pile driving is one that's pretty easy to mitigate, actually, but the others are really affecting marine wildlife and also fish. Good. So in my group here in Denmark, we study basically bioacoustics, so sound communication and behavior in our group, in in a context and in the interface of biology, acoustics, physiology, robotics, and neuroscience. So it's a rather broad group. We have now 7 PIs, or about like 30 people or so in total, and so it's really fun, nice group. And my focus has been of this whole sort of array on the production side. So how do animals produce sound? And we have other people who are interested in other people interested in perception, for example, or sensory motor integration. Good. So my main research focus has been how does brain and body interact to produce voice? And we do this at different time scales. So we look at immediate time scales. So how does it neural control of the system? We also started looking more and more over developmental time scales. So how does during learning, for example, and also at evolutionary time scales, how do have different animals evolve different structures to produce sound? Good. And I notice I'm preaching to a little bit to the choir here for this audience, but it's actually super nice. So if you're interested in behavior, right? So one of the major outstanding questions in neuroscience is how does the brain produce behavior? So how do you go from birth to behavior? And if you do that, I really like to think of this in this embodied concept where you have the brain as a neural controller that sends motor commands to the body, and in this case, first muscle, of course, where you have action contraction coupling that then leads to forces that change the motion of the body, that then again leads to forces and torques on the environment. Now, there's lots of mechanical feedback loops that you know of very well here in this audience, like proprioception, but there's also environmental to body. For example, when you're swimming, you really feel the fluid, or flying, right? You feel the force of fluid dynamics, but also intrinsic properties of the muscles that, for example, length velocity dependencies or stretch activation. Now, this you all know, but I think it's always important to say, and especially if you consider that typically the people who do these different, or work on these different problems are in different fields, right? So the neuroscientists are typically not studying so much biomechanics or much behavior. And I would like to pose, and I think here you all agree, that if you want to really understand the neural basis of behavior, you need an interdisciplinary approach where you try to integrate these systems. Good. So that said, I'm interested in voice production, and voice is really an interesting problem because it basically it's a very complicated system to control. And just to illustrate that, I love to show this video always from Sarah Lundberg. What we're going to look at is 4 people singing for you, and they're all going to get an endoscope. They're getting right now getting an endoscope through their nose, and what we're going to try to do is image what happens in their larynx when they sing. And so this happens to everybody you talk to all the time. It's happening to me right now. So it's a really absolutely gorgeous system of fine motor control that involves hundreds and hundreds of muscles that we just don't see, right? It's not like locomotion where it's so external. Okay, so what you see now here is the two vocal, I hope you can see my mouse. This is the two vocal folds. The big thing here is the epiglottis that protects airways from food. This is regular breathing. And now we're going to get sound production here. We have reduction of the vocal fold. More. And a bit. Good. So what happens when we produce sound this way is that we basically bring the vocal folds here in red together by movement of these purple cartilages called erythroids. And then they start oscillating. And in this video is this 25 hertz frame rate video, so you can't see the oscillations because they're at hundreds of hertz. Good, so but at least the larynx, where the sound is produced, is a super dynamical system where you really like have lots and lots of motions, very precise control over vocal fold length, positioning, and so on. And this happens, we're all experts at this. Now, then you have another motor system which modulates the sound that's produced, and that's in our case the upper vocal tract. So that includes things like the tongue and the lips. And here you see an MRI of somebody beatboxing, and just appreciate the shape of the tongue, right? The tongue is so cool. It's incredible. There's the larynx in the bottom. Look at that tongue. It's so cool. And here's the palate moving fast. Good. So this is a great excuse to put people on MRI machines and making them beatbox. But basically the idea is that if you take the same embodied principle and now apply it to voice production, you actually have to deal with three motor systems. So you can have a brain that's the song system in songbirds, for example, but it's now mapped on three systems. So one is a respiratory system, where again you have muscle, you now have a skeleton that drives the dynamics of respiration that produces a driving pressure, for example, in the trachea in our case, that then maps and is another it maps onto another motor system, which is the larynx or the syrinx in songbirds, where again you have muscle, the motion of the skeleton of the syrinx or larynx, and then that interacts with the sound production mechanism that then causes fluid fluctuations. And these go to the next system, which is then the upper vocal tract, where again you have muscle, you have now the skeletal dynamics of the vocal tract, which typically filters or radiates, it changes the radiation of the sound, and that results in the voice that comes out. Now, there's lots of feedback loops in this system, but it's yeah. So when we put this together about 10 years ago, so it really was really very insightful for me to say, okay, what's really critical in this part here is to understand the sound production mechanisms and understand how they are controlled. So which control parameters do we actually have? Good. So we need identification of physical processes, but also the control parameters of sound production before we can even start thinking about all these other motor systems. And that has sort of really guided me in the last 10 years to look at a very comparative way of identifying these sound production mechanisms. Now, what we know in humans is that we have vocal folds that vibrate. They cause flow modulations in the vocal tract, right? That's how we produce sound. Now, this idea is very simple in a way. It's called the myoelastic aerodynamic theory of sound production, and it sort of counteracted the idea that every sound pulse was made by muscle contraction that people thought around in the 50s. And the theory development was really driven by people interested in modeling the system and compressing the sound into model parameters instead of transmitting the sound itself. And this is really at the start of the telephone industry. And so Bell Labs, for example, played a crucial role in this in the 50s. Good, so we have vocal folds that oscillate and are flow-induced self-sustained oscillation. So you have basically an interaction between the airflow passing them and then configuration change between opening and closing that and elastic forces of the tissue that make this a self-oscillating system. And you can really nicely compare it to like people pushing a swing, right? If you want to get the swing going, you need to just push in the right phase and you inject energy in the system. And if you do it in the wrong phase, you would break it. Good. So it's actually quite complicated physics. So it involves fluid mechanics, of course, but also soft tissue motion. So not rigid bodies, but like floppy things that have huge excursions, which make it really hard for a lot of computational approaches. And then acoustics and often interactions between these three different types of physics. So it becomes quickly rather complicated. And in terms of getting data, it's high-speed events often occurring hundreds of times a second or thousands of times a second. So you quickly need filming to observe this at thousands of frames a second. Now, this is difficult in humans and let alone in laboratory animals. So if you look at the cameras we typically use is one of these photon cameras that can go very fast. This thing's 15 kilos and trying to get it in like the model systems like songbirds or mice or rats or so, it's just really, really, really challenging and making them vocalize, right, while they do this. Good. So, and there was a lot of colleagues like Franz Goller and Ola Larsen who really dedicated like 10, 15 years of their life to film, for example, birds while making sound. And it was so difficult. And so I decided to take a different approach and a more in vitro based approach and taking the organs out instead of trying to do it in vivo. And so what we developed basically is a program where we can take organs out and just study basically by blowing air through them how they produce sound. That's one thing. But then I was also trained doing muscle physiology. So we did a lot of muscle physiology on these different muscles. And then I started combining these two, like the sort of knowledge of these two systems into setups where we could, sorry, control pressure, keep the systems alive, stimulate muscles, and then study the effect causally, right, of the muscle stimulation to the sound production. Good. And so this is an example, some data. So this is started like 10 years ago or so. This is what you see here is an image of a zebrafinch syrinx oscillating, where we could annotate some of the vibrations, slow down like 300 times. So this was filmed at 35 kilohertz or so, and then the oscillations happen at 500 to even higher, 500 hertz to higher. Good, so that's the kind of data we could produce. And since then, we've just been doing this in lots and lots and lots of different animals. And I just wanted to highlight a few findings of of this last period, basically. So in a lot of animals, we see normal laryngeal function as in normal as compared to us. So we see similar function as in humans. But we've also found, for example, in marmosets or bats, that there's very strong adaptations. And in both of these animals, there's very strong adaptations to make very high frequency sounds. So we found, for example, that in bats, they have these long extensions of their vocal folds that are very thin membranes, basically. Now, bats are really, really fascinating because when I started thinking about this, I realized, if you look at the range of frequencies an animal can produce or human can produce, it's typically like 3 to 4, like it's 2 to 3 octaves. Like a good untrained singer can do 3 or so. But every bat does like 7 octaves easily. And so the best singers in terms of vocal range, like people like Mariah Carey or Axl Rose, they can get to 5 or 4 octaves, right? This is really the extreme what humans can do. But every bat can do like 2 more. And so we were really interested in how do they do this. And it turned out that they use these very thin vocal membranes for the very high frequency ultrasonic calls that they use for echolocation and for navigation and catching insects. And then they have a second set of tissues that they oscillate for much lower frequency communication. And these are the false vocal folds. Good. And so we have basically completely functional now. They use these things for very different things. One is high frequency sounds for echolocation. These sounds are very directional, so they're really bad for communicative signals because you basically have like a flashlight and point it very focused at one direction, which is not good if you want to attract attention, right? So these false vocal folds have adapted for the much lower frequency sounds that are omnidirectional, and they use them for communication. Now, the only other animal we know of in this time that actually also makes sound with their false vocal folds is death metal singers. And so when this paper came out, it was absolutely hilarious what the what we had a media attention. And so Will Ramos, for example, from Lorna Shore, he's this more like modern death metal guy who made this fantastic YouTube video of like 20 minutes about the paper itself, which is totally awesome. Really, really great. Good, but so there's these adaptations. Then we also see that mammals like mice and actually a lot of rodents, we think, and also xenopus have evolved completely different ways of producing sound in the larynx. I won't go into too much detail, but for example, mice and rats, we've now convincingly shown that what they do is they put their vocal folds together, and that leaves a very small opening that's left. Then you get an air jet that's forming thanks to that small opening where the velocities are getting so high, you're actually getting into like 10% of the speed of sound. So subsonic jets, and these are pointed inside the larynx against the wall of the larynx, and these jets basically get interaction with the sound that's produced, and the sound is then stabilizing vortex shedding. And this is very similar to, for example, these Harrier aircraft carriers, where they land, or these are these vertical landing planes, or and they basically, when they point this like supersonic jet downwards, you can also get these very strong whistle tones because there's a stable waveform that's going traveling back into the jet. Basically, that's the sound. Good. So that was completely wild to find that that's the mechanism of sound production in basically mice and rats. So it's very different from us. Good. And then we found the same mechanism of vocal fold oscillations, but in totally novel organs. So we've shown in birds now very convincingly that they use this exact same mechanism in a different organ, the syrinx. I'll come back to that more. But also we found that the odontocetes, so this is half of the whales basically, this is the killer whales and the sperm whales and all the dolphins, they produce sound with structures that sit sort of high up in their nose in a completely new location. And then a little bit over a year ago, we also shown that in the other group of whales, so this is the very large baleen whales basically, they still produce sound with their larynx, but with a totally different structure in their larynx that's adapted for that only whales have basically. And for this audience, I decided not to go into any of the whale papers because I think I wanted to tell you something, show you something else. If I have time left at the end, I'll maybe do this, but I don't think so. I will. Good. Okay, so basically, human speech laid really the foundation for the study of animal voice production. And we've shown over the last 10 years, together with other labs, that this meat mechanism is really universal across the tree of life. And the tree of life here is basically the vertebrates, I mean, there. So it's not the entire tree of life, but it's the vertebrates that produce sound. And by doing this basic research, we deliver really good data, actually, right? So really, we can image at very high speed. We have very high resolution experimental data. And funny enough, the human voice field actually lacks such data and such possibilities to the experiments we can do. So it's gone sort of full circle, and I think this is very interesting. And we're at a time now when I think we can start really informing the human voice field. Now, and one of the things we've been doing is developing computational voice models that have such level of detail that would be very hard to validate in humans at this point. But these models could really move things forward in terms of understanding structure-function relationships, understanding the motor control or evolution or disease models. Great. So what Simon briefly introduced also is that over the last 4 years or so, 5 years, we've been working together with a large group on basically making a model where we can go from bursting to song, right? So really capture sort of the complex motor control of the system. And there's really a wave right now, I think, of this in different systems. So also like in locomotion, for example. And one of the things, of course, that really triggered this is that we now have really amazing tools to do animal pose quantification, like Mackenzie Mathis's Deep Lab God or Talmo's Sleep, and there's other systems there, of course, now that really change the way people can quantify motion in a fantastic way, right? And then the other, in parallel to that, is that we see massive development of like large arrays of recording animals. And what's really fascinating to me, in just the last few years, we see that people really started producing completely virtual animals, like so really like animals that are driven by sorry, that where people are really getting interested in biomechanics. Good. And the point I wanted to make with this, and of course the other group is the people in Janelia are doing amazing stuff with the robo flies, or neuromech flies it's called right now. Because I think the main message I want to say is hurrah, because it seems that finally systems neuroscience people care about biomechanics, which we really care about. And so I think this is a fantastic movement that's really now happening in sort of neuroscience in the last few years. Of course, there's been people around who cared about this more longer than that, but there's really a movement, I think, and that's very exciting to see. Good. So in this movement, we're also hopefully being part of that, and we're trying to develop models for control of voice. Now, if you look at the human voice modeling, where that really started in sort of the 1950s, this is a cross section of vocal folds of humans, basically. So you have basically this is the part that oscillates, and here is a muscle that controls the tension to some extent. And this system has been approached in various different ways. So you have these models let's say on the left side, we start with extremely simple, computationally simple models, where the whole vocal folds have been reduced to two masses or even one mass that's flopping around, and so really reduced order models. Then there's been people who've made more realistic, more physics-based 2D or even 3D models of the vocal folds. And so the highest end in terms of complexity we have now is a like a fully articulated dog larynx, in this case actually, with a human vocal tract on top. And I just wanted to show you this is sort of the state of the art now. So this is really all these different colors here on the left are different muscles, and they're activated. And then here's the oscillation, which then couples into the vocal tract. Good. So this is really, really fantastic. I mean, only like a few years ago, this took so much computational power to do this, and it's moving, of course, very fast. But this dog, larynx, with a human vocal tract, can now say this, which is still very far off from, of course, actual speech, but it's getting there. Good, but I guess the difference between these models is that, like on these very simplified models, is that they're very fast. They can describe a lot of dynamical features of these systems, and they allow you to do very broad parameter space explorations. So that's really, really nice. But one of the cons is that it's very hard to relate these to actual physiological representation of the materials, for example, or the geometry or muscle activation. So that's just much more difficult. And these really high-end, high-fidelity models have this representation of physiology, of tissue properties and geometry. They could actually link vocal fold properties to sound production in anatomically correct models. They're just super computationally expensive. You can do very limited parameter exploration, and you need detailed parameterization. I mean, of course, you've had exactly these approaches in locomotion field as well. And I guess the main thing is like, what's your goal, right? What do you want to achieve with the models? And so in the song field, people have really looked at these sort of reduced order models for many, many, many years. And I thought like, I really want to start developing these high fidelity models because I know there's these limitations, but there we can really, if it works, we could really move, right? We could really start to look at like things like evolution or like structure function relationships. Good. And so we needed to come up with a way to sort of this model that I presented earlier to capture this in a computational model. And the animal we focus on is a zebrafinch, which is a songbird that's really heavily studied. And one of the main reasons for this is that this is a cross-section of the brain. I won't go into too much detail, but in the United States alone, there's about 140 labs that study just the neural circuitry of the behavior of this animal, which is learning to sing in a way that's very, very similar to that as humans learn to speak. So like the statistics of babbling and so on, there's so many parallels. So this is one of the really beautiful models of a learned behavior of imitating sound in a higher vertebrate, where we really know very well what the different functions of different brain nuclei is. But as you can see, this is the brain. Here is sort of the hindbrain with the motor neuron areas are here. And so basically there's always this arrow saying like towards the trachea and the syrinx muscles. And so that's where the neuroscientists typically just stop, right? And going back to my original thesis, I think to really understand the vocal production, we need to integrate this whole part of the biomechanics with the neuroscience to try to understand how the system is controlled. Good. So over the last years, we've been starting to fill in a lot of the details here. And this is the vocal organ of a songbird. It's, a zebrafinch is about 15 grams. So this vocal organ is about 7 millimeters or so in height. And on those 7 millimeters, there's a lot of action packed. So each side, so here you see the trachea in yellow. It splits towards the bronchi, towards the lungs. Now, in songbirds, you have a pair of oscillators that sits in each bronchus. So songbirds have two sound sources that they can control independently with our left and right hemisphere, completely independently. Each side has about 8 muscles, depending who you talk to, but they control position and basically different parameters of these vocal folds. But of three muscles, we actually know what they sort of do. The others are still quite mysterious, and it's very hard to study this with EMG in a singing tiny songbird. Good, and this is really a lot of work done by Frans Goller and Rod Sudders and Ole Larsen in the 90s and 2000s. Good. So we wanted to go to build this system into like a high fidelity syrinx model. And I've done this together with a great group of collaborators. So I have collaborators in the Rochester Institute of Technology in the USA, so Xiang, Sudong, and Wei Li. And then Sam Sober at Emory at Atlanta. And then my team in Denmark with Iris, Adam, and Nick Ladman, Peter Rafalt, and Maria Antonsen. Good. And so just to give you the layout, I won't go into every detail here because it's too much. But basically, we needed to set up a set of simulations, right? So we needed to be able to predict force from spiking behavior of neurons. We then needed an actuated skeleton. We needed to have the correct soft tissue properties and models that could capture the vibration into finally like this fluid structure interaction model of the sound production itself. Good. And on the experimental side, that was our part of it. We needed to establish lots of different material properties like muscle properties, non-isometric muscle properties. How are the motor units distributed? How is the activation of these motor units during in vivo behavior? And then also try to figure out how we can 3D motion capture this tiny organ, preferably in vivo, that we never managed. But even in vitro has been extremely challenging. Good. So again, as a thread here, I'll go to a few of these. So the first sort of problem we had is like we need some muscle model that can go from spiking behavior to force. And the muscles we're dealing with, and I've been working on for many years now, is it's an interesting set of muscles because they're a group of muscles that we call super fast muscles, and they're it's a group of very fast muscles that we found are ubiquitous in vocal communication. So there's toadfish, the epic work by Larry Rohn, for example. We've established them in birds, but also in bats as mammals. They're basically the fastest synchronous vertebrate skeletal muscles possible, right? They can move things. They still produce positive work and power up to 275 hertz of cyclical motion. And in this very comparative study, 10 years ago or so now, we looked at all the different superfast muscles that are out there and basically realized they really have converged in different ways upon extremely fast calcium handling or like packed with mitochondria to deal with all the fast cycling and the high energy amounts needed. So, but you can make a muscle much faster, we think, because you just get so many membranes on the inside that you don't produce enough force anymore to overcome that internal elasticity. So then we've shown that the muscles are so fast that they can actually execute millisecond precise spike timing. This was worked together with Sam Sober, where and actually I should say that that goes. They're so fast that actually the reaction time, the activation time of the muscle hits the refractory period of the motor neuron, right? So then that order of magnitude, like 1-2 milliseconds, full force development. And then another cool thing we found, I really love this, is that one of the problems we had when recording activity is that we just get so much activity during singing, for example. And now we figured out why. So I started this super simple project with two bachelor students. It's like, let's just count like how many motor axons are there actually, and how many muscle fibers are there, and can we just then see what sort of distribution models, like how big are the motor units and these kind of things. And from these models, we predicted that like 30% of the muscle fibers actually was innervated by a single motor neuron. And so we submitted that, and I thought like this is impossible. And then there was also the reaction of the referees who said this is impossible. You need to show more evidence. And so we developed this cool prep where we basically had the a motor nerve and the muscle intact, could stimulate individual axons, but could then image a whole cross section of the entire muscle where we had calcium indicators, and so we could see basically on a confocal which of the muscle fibers were activated by which which motor nerve. So that was really cool, and we could show actually that they have this incredible high control resolution where like at least let's say 13 to 15% of the muscle fibers is innervated by a single motor neuron, which is weird invertebrates, right? Good. And then more and more we start to realize that these are actually part of a group of craniofacial muscles with all kinds of other adaptations, but I won't go into too much detail there. But anyway, we wanted to capture all these behaviors in a muscle model. And this image is very familiar to all of you in the center, so I won't go into too much detail. But the challenge we had is that we wanted to drive it with naturalistic behavior. So sort of a hill type activation model we considered too simple because it didn't allow us directly to put action potentials in. But then if you then go to like a Hodgkin-Huxley model where you have the whole excitation contraction coupling, we just got it's just too computationally intensive and we had to fit so many parameters that we basically never had, right? So we chose a sort of in-between approach um, where we do capture the basic processes happening during excitation contraction coupling and where we were allowed us to put in multiple spikes with different timing variability and so natural recruitment, right? What you see in an animal. Good. I mean, we named this model Yisuji, and this is a sort of which means, I mean, this is a Mandarin, which is such a beautiful language because this literally means fast, super fast muscle model. I thought, oh, simulation. So simulation, really great. Good. And so what we basically do in this process. Here you see an overview of different, basically, force responses to different stimulation patterns. And the patterns you see, for example, here on the top. So this is like 5 different patterns in a row where we just subtly vary the timing of these pulses. And we did these experiments, and then on a subset of the data, we trained the network like to fit the model. Um, on the parameters we have in the model, and then we predicted basically the other set. And overall, we get a really nice correspondence between the prediction on the y-axis here and the experiment on the x-axis for different parameters like maximum force, area under the curve, or the timing to the maximum force. So this was, there's always improvements on these models, but this is at least a very good step for us to as a muscle model. Good. Then another major thing we needed to deal with was this motor unit distribution and how did this muscle actually getting used in vivo. And I just want to briefly highlight there what we did. So in this paper that I just also briefly mentioned, we managed to image the motor unit distributions in single individuals in a single muscle. So this was really nice. Instead of like using lots and lots of different mice for screening and making one connectomic map, we could actually do this within a single individual, but only very small muscles. Good. Then the other thing we've been working on for a very long time together with Sam Sober at Emory is to make new electrodes. And this has really come out of the need for Sam and me to record songbird vocal muscles. And Sam has completely gone on the electrode bandwagon. So in the end, he basically produced these very beautiful, basically printed devices that are implantable, chronically implantable, and he's doing it in all sorts of different animals. But it's really come out of this, out of the songbird and need. So this is really, really exciting. And these allow us now for the first time to record single unit activity in songbird muscles while singing. It's still extremely challenging, but it does work. Good. And then I'm going to skip all these other things except for 2 more pieces and then I'll show you the end. So basically we went from a very coarse morphological anatomical description of this vocal organ to a much finer one using a synchrotron where we basically now have a single muscle fiber map of the whole organ. And we could then implement it into our actuated skeleton where we can now simulate basically, the activity of the muscles. Now we're validating this in vitro preparations because in vivo has been too difficult, where we can 3D reconstruct basically the small organ while we stimulate it under a stereo microscope and validate these measurements. But sort of the end here I wanted to show you now is that now we have a syrinx. This is one side of the syrinx with all the muscles attached to it, and we can blow air through it, stimulate the different muscles in different regimes, and then calculate the flow that comes out and the sound produced and so forth. So this is like a short simulation of that, where we activate this muscle here on the front that is controlling fundamental frequency And so this is really like a major step forward for me and also for the field, I hope, for in modeling this system. Good. Now I have a few minutes left, and I I think I want for this audience I wanted to uh, talk about one little thing for a few more minutes, and that's a paper we had in the last few years, but it's really I think changed the direction about how we think about actually vocal performance, and that really ties into locomotion performance and exercise. And that is that we found that vocal performance also needs exercise. That's the bottom line. Um, Birdsong is used, you know, in science heavily as a great model system for vocal learning in general, but also like human vocal learning, which is the capacity to imitate sounds. And I briefly mentioned that already. But if you take a zebrafinch, as it develops from day one to about 100 days as an adult, in this short period of time, it learns to sing by copying its father's song. So we now know that, for example, in the tropics, both males and females sing, but a lot of this work is still based on the species where only the males sing. And here we go. Good. And the beauty of this system is that there's different phases of learning. So first the birds listen. There's this phase from like 15 days to about 35 days where they just listen to the adults in their environment. And then separate from that, they start to practice. And that's these two phases of auditory learning and sensory motor learning are kind of decoupled in most species. And they produce first subsong, which is they sort of get the timing right, and then they start to get the frequencies right when they move to plastic song. And in this species, it crystallizes to a song that these animals will sing for the rest of their lives, hundreds of times a day, with a variability of like 2%. And in this one, it sounds like this. And you get the beauty here because we can basically record this behavior extremely easily. We don't need video cameras. We just record the sound and we can record every single sound a bird makes during this whole development. So where we record like 5, 6 million syllables of an animal over this. So we can record everything. It's practicing, right? Good. Now what we found is that over the song learning phase, we saw actually the muscle speed is increasing. So it ends with being like this extremely fast muscle, but it doesn't start there. And we thought, is this development? Is this hormonal? Because we don't see it in the females. Is it exercise? And by just reviewing literature, we could actually rather quickly exclude hormones. But hormones have an effect, but it's not massive in this species. And so it was, is it development or exercise? And and this is beautiful, I think, because it's really in a phase like lots of different behaviors we learn that happen exactly when we also grow, right? So like this, I think I love this image because it so captures the skill we develop and the body we develop over when we develop, like so when we grow into adulthood, and how much better we get at precise motor control. Good. So one here you see a syrinx from on the left side a juvenile and on the right side an adult, different individuals, obviously. And you see there's a massive change. So the overall size of the vocal organ is the same, but the muscles here are just they're much bigger and much more developed. And so the first thing we did was a very crude experiment. We basically just did a nerve cut on one side. So this is the beauty of the system. It's completely bilateral symmetrical. So the birds still can sing on the other side. They won't use that side if you do a nerve cut. And what we saw is that the muscles basically on the nerve cut side just stay in this juvenile stage, while the ones that are used do grow and develop completely normally in an adult. Now, so we did these nerve cuts as juveniles and then waited 70 days and basically looked. So we have an internal control, right, which is nice. Then we looked at force development. Here you see stress over time of a single twitch. And as a measure of speed, we just use the full width half maximum, so the twitch half width basically. And we see that as juveniles, it's about 8 milliseconds. And as an adult, it goes to 4 milliseconds. This is incredibly fast. Both are fast. But if you do this denervation, we go from the 4 back to 8, basically. So that's the speed. If you look at the force these muscles produce, so this is the tetanic contraction, and we look at the maximum isometric stress, we see that it's like a 10 millinewton per square millimeter, and it goes down to 5. If you then do the nerve cut, it basically goes back up. So our conclusion here was that muscle use is needed during development to acquire the speed. So this is during development. And we did the same experiment in adults that already sing and already have fast muscles. And we looked basically after 2 days and 21 days what the effects would be of a nerve cut. And we see in terms of speed that there's already after 2 days an effect, but it's not significant. But then at 21 days, these animals are basically back to the level of a juvenile. So also as adults, they need to use these muscles to maintain the performance, right? Now we did a lot of different work. So we looked at, anatomy. Here you see cross section, for example, of the left and the right of the syrinx, with one side nerve cut, and here you see zoom in. So here we see massive shifts in fiber typing. We looked at the proteome. I won't go into too much detail, but basically we see massive shifts in all these different things due to nerve cuts. Now, nerve cuts are really unnatural things to do, of course, but it was sort of the hard line experiment we could, or like the extreme experiment we could do. But what we were really interested in is like, okay, what is now if we just change the behavior, right? So if we just can we prevent them from singing? And in the end, we did. So we could prevent males for singing for about 7 days, or we did it for 7 days. And we did this by putting them in the dark. So we also changed melatonin in that sense. But we have controls for that. But basically, the animals were just not singing. Otherwise, they were completely doing everything rather normal. They were sleeping more. But what we saw is that the singing also had a strong effect of muscle speed, also changed the acoustics. And so in lots of different birds, it changed in different ways, but it changed the acoustics. And then we did the experiment, which I really like in this paper, is that we asked the only animals that care about this song, that's the females. So we asked the females, if you now have a song from a male that's trained normally and then didn't sing for a week, which one, not just which one is different, but which one do you prefer? They completely picked out the ones that before they were detrained, basically. So they preferred the song of individuals when they're at their peak, basically, right? Good, so there I wrap it up, but I think this has massive implications because one of them is that it implies that birds need to sing a lot to exercise their muscles and to maintain this performance, and that it matters in a sexual context, right? And that means that voice for these animals actually contains information. The females can extract this information on exercise investment. So how much have these animals trained? And that is another consequence because it's a completely novel explanation why songbirds sing every day, right? Right now in the morning again, in the States as well, you can hear birds are starting to sing again. And there's lots of different reasons people have come up why birds sing every morning, but they're all not very convincing. And this is a completely novel explanation. They need to just exercise in a time period where they can do it and it's when they still cannot forage. And it basically couples the field of exercise physiology to vocal performance. And it's so obvious in retrospect that this makes sense, but just nobody's thought about it. And so this is really beautiful because now we can really tap into this massive knowledge we have in exercise physiology and see where this is applicable to vocal performance and where not. Now, and one thing I want to end with is that if you look at sort of limb muscle in vertebrates at least, if you train them, you can train them lots of different ways, of course, but overall, if you do strength training or endurance training, you see that there's effects on the fibers. But typically what happens is that they go from fast fiber types to slower fiber types due to exercise. And disuse or like, different disuse paradigms change these slower, but like, for example, more fatigue resistant or stronger muscles to faster muscles. While in these syringeal muscles, it's the exact opposite. So we see if these muscles are already very fast, but if they use them, they get super fast, and you see that the force goes down, their fatigue resistance goes up, and disuse switches them back to fast. So they react strongly. This is really a novel finding, but then they do it in the opposite way, basically. And so this, I think, really opens up a whole box of questions we're going to now look into. And this is also applicable for humans, for example, right? So can we apply the knowledge we've now found in songbirds, where these muscles are probably very conserved, to humans in health and disease? Can we look at singing pedagogy, where we know there's lots of training happening with human voice, but there's a great lack of sort of mechanistic insight of what this actually does. We know it helps, but we don't really know why. And also an aging voice. So how does muscle control changes over aging, and how does it affect the voice? Good. And there I would like to stop. I would like to thank my group and lots of different funding agencies for funding my work over the years, and you for your attention.
That was great, Coen. Thank you. So questions?
Yeah, I have a question. Um, so I come from kind of like a more of a locomotion muscle background, and I would think of sort of the function of a muscle involved in locomotion as being highly dependent upon the phase of activation in relation to the locomotor cycle.
Yes.
And in particular, when you change the phase of activation in vitro, you get a change in work production of the muscle, and that's going to affect motor performance. And I think, obviously, I don't know very much about vocal performance, but it would seem to me that if you're modulating frequency of your voice constantly, then there's going to be some frequency modulation maybe somewhere in the system. So I guess my question is, what's the relationship between kind of precise control of phase of activation with respect to vocal performance? Is there a relationship? How complicated is that relationship?
Yeah, so the very quick answer is that we just don't know yet. So basically, we have very little EMG data. We have almost no muscle shortening data, like muscle length changing during singing, because nobody's managed to measure this precisely. So, and that's a great difference with like locomotory studies, right, where these kind of metrics are available. So we don't know the actual length changes, and we don't know the really nice the activation yet. But that said, so the a lot of these behaviors are not so cyclical as locomotion, right? So they're very precisely controlled in terms of like muscle tension, for example, or like sort of vocal fold tension, for example. And I think a lot of it is there's few antagonists also. So a lot of it is this sort of the passive elastic components that pull things back into shape. And then let's say one muscle will control the frequency by tensing a membrane. And if it's deactivated, it will just go back like passively. So I mean, I'm very aware of that. And I know those papers very well. But we're not in anywhere close to look into that. Like what I said, like with those 8 pairs of muscles, some of them I think could function as stiffeners to stiffen this whole system up. So to change response times, for example, there's a few that could be antagonists, but we're starting to now systematically study this by stimulating them one by one, seeing their effects on not just acoustics, but actually on the geometry of the skeleton. And then the next step will be the acoustics.
So, Coen, I think Ethan asked a very maybe more articulate version of the question I was going to ask, but it launched something for me. I was going to ask, like, how do you get the dynamic, where are the sort of levers of control that give you the full dynamic range of sort of song production? And I guess maybe my more focused question is, do you think there's bandwidth separation that's significant in this system? And where is that? Is that between the biomechanics and the muscle control, or between different muscles in the system? Where's the sensitivity coming in, and where's the insensitivity coming in?
Yeah, I mean, so again, like we don't really know yet, but what we do know is that sort of the oscillator, which is producing the sound, is highly differentiated between left and right in most songbird species. So, for example, the left side typically produces, let's say, 500 hertz to 3 kilohertz, and the right side goes down. And so the animals have to learn to switch hemispheres to make a perfect glissando from high to low frequencies. And that is gradually basically gating the sound in one and the other, and they learn to sort of make this completely smooth, make this transition smooth. But like in terms of so when a lot of it is done by the biomechanics, right? So a lot of the sound that comes out is really set by the material properties and the interaction with the airflow. But one thing I'm really intrigued about is that this system must be super robust to perturbation, because basically these oscillations are very large in motion, and the muscles are being sort of jerked along with it. And but they can occur at like an order of magnitude faster than the muscle actually contracts itself, right? Then there's the heart sitting next to it doing boom, 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 boom. And so there must be some resilience against perturbation in the system. And we just haven't studied this yet. Like I really love to apply these concepts from the locomotion work to that.
And I guess an extension of that is, how much do you think, if if all your dreams came true and you figured this out really well for the songbird vocal production, what aspects of it do you, given your really cool comparative insights that you were talking about, what aspects of it do you think are most likely to generalize, and what aspects do you think are really sort of going to be very system specific? I think this is partially my ignorance on the different sort of biomechanics of the syrinx versus the larynx and all. I mean, I see the difference, but I don't know how that manifests
No,
dynamically.
that's a great question. I mean, so I think in terms of muscle and yeah, basically the muscles themselves, they I think are all craniofacial muscles. We have to still show that conclusively. They respond differently to training in ways that are also not well known, also not for extraocular, for example. I think that's very translatable, because the myosins that are involved, like MI13s, are really ancient, right? They sort of predate even splits between birds and mammals. Like, I mean, they're old. But we don't know that very systematically. But I think that's very, very applicable broadly. In the sound production, dynamics, for example, is surprisingly robust. It's super cool. So, for example, like 2 years ago, we showed that, for example, all the dolphins, they make these very distinct sounds, and we have really good evidence now. What they do is actually exactly what we do with our voice. They shift into different voice registers. Like from the normal chest voice I have now, I can do falsetto, this one, and then you go to a different frequency range. And these animals seem to do that too. And the dynamics where they switch is could be complete coincidence, but it's exactly at the same sort of opening closing ratios between humans and dolphins, right? In a totally different system. And in songbirds, nobody's really looked yet. It's probably so there are some very basic parts of the nonlinear dynamics that are really translatable, like bifurcations, like period doublings, or to deterministic chaos. They seem to really occur in very similar ways.
Okay, well, I don't want to monopolize the questions, and folks are having to start taking off because we're at the hour. I want to open it up if there's any other questions from other folks, since we started a few minutes late.
Quiet.
It's always hard on Zoom, right? We have this recorded, so we'll post this to the IMSI website and have follow up. And I think we'll post Dr. Elemans' contact information as well for everybody to follow up on. I guess it's in your slides too, so we can do that. But I guess I'll ask one last question then along that, which is, so along those lines, are there non-dimensional numbers and such that can capture some of this that you think across species as a sort of extension to that? Because that's a really common way of thinking about where, how we would scale properties of like the biomechanics and other systems. I just don't know if there's ones that exist yet.
Well, I mean, a lot of the sort of the things related to fluid dynamics apply. So but so actually people haven't looked into that so much in terms of voice sound production, much more into whistle production, where that's really heavily driven by fluid dynamics. So there you can see, it's certain like strong numbers, or so you get transitions to stable whistles or so, but it's not systematically done here actually. Also, I think one of the reasons is that we only until very recently didn't have access to a huge size range. Like everything was done in sort of human-sized animals, but now we've studied like mice and songbirds, and then now we have, whales. So we have like a seven orders of magnitude in weight that we can start looking into.
All right, we'll check back in once you got the sperm whale analyzed.
Yes.
Let's thank Coen again for a really excellent talk, and thanks for doing this again, especially at a time that's inconvenient for you. So
No
thanks
worries.
very, very much.
Good. And I hope to meet you somewhere all at a neuroscience meeting or wherever. Great. Take care.
Thank you.
Thanks again, Coen. I'll check in with you tomorrow about stuff, or maybe the next time out, because I do have a bunch of other follow-up questions. But that was really, really great. Thank you so much for doing that. Oh, maybe he's not here anymore.
Yeah, thank you.